We are diving past the protective sheets of a human nerve bundle, past the individual fibers, down to the microscopic cellular membrane. Here lies a major biological engineering problem. The human body has to coordinate instantaneous survival responses, but it has to do so using wet, highly resistive biological tissue. If the body relied solely on chemicals diffusing through fluid to send a message from your toe to your brain, the delay would be measured in minutes. Conversely, it cannot rely purely on electricity. Biological tissue doesn't conduct current like copper wire. A purely electrical signal would bleed out into the surrounding fluid almost instantly. Neurons solve this paradox by alternating between two distinct methods, using internal electrical gradients to travel rapidly along the length of a single cell, and utilizing chemical messengers to cross the physical space between them. To see exactly how this works, we are going to build the electrochemical model of the action potential from first principles. We will trace the exact sequence of events, starting at the resting lipid bilayer, tracking the initial voltage spike, riding that impulse down the axon, and finally crossing the synapse. This specific electrochemical sequence governs basic physical reflexes and determines the lethality of various neurotoxins. Before a neuron can fire, it sits at a resting membrane potential of precisely -70 millivolts. This internal negative charge is dictated by a strict environmental setup, a high concentration of sodium ions outside the cell and a high concentration of potassium ions inside the cell. This diagram shows the cellular membrane, where a specialized protein called the sodium-potassium ATPase actively pushes 3 sodium ions out into the extracellular space and pulls 2 potassium ions in. Because it moves these ions against their natural concentration gradients, the pump requires constant energy, spending a massive portion of the body's total ATP to function. But the membrane is also dotted with passive potassium leak channels. These are always open, allowing the positively charged potassium to slowly drift right back out of the cell. As positive charge leaves, the inside of the cell becomes increasingly negative. If potassium were the only ion moving, the electrical pull would eventually balance the chemical drift, causing the voltage meter to plummet toward -90 millivolts. However, a small amount of sodium leaks inward at the same time, attempting to drag the voltage up toward +60 millivolts. The cell balances this ongoing chemical tug-of-war, and the voltage stabilizes at precisely -70 millivolts. The neuron acts as a tightly coiled spring. It consumes energy to hold these chemical gradients at bay, storing potential energy for an instant release. That release is calculated at the axon hillock, the region where the cell body connects to the outgoing axon. Here, the neuron constantly tallies incoming excitatory and inhibitory signals. If excitatory signals arrive rapidly enough, or from enough simultaneous sources, the local voltage begins to rise. The voltage creeps up from the resting baseline until it hits a critical threshold, usually around -50 millivolts. Once it reaches that precise mark, the action potential triggers. There are no partial signals. It is a strict all-or-nothing response. This graph charts the exact sequence. Hitting the -50 millivolt threshold mechanically forces voltage-gated sodium channels in the membrane to swing open. Because sodium is highly concentrated outside and the cell interior is negatively charged, the ion is driven by both chemical diffusion and electrical attraction. It floods into the cell. This massive influx of positive charge causes the membrane potential to rapidly depolarize, spiking well past zero to peak at +35 millivolts. At exactly +35 millivolts, the physical structure of the sodium channels changes again. A mechanical plug swings shut, instantly inactivating the channel and stopping the sodium influx. This extreme positive voltage simultaneously triggers adjacent voltage-gated potassium channels to open wide, forcing potassium to rush outward. As the positively charged potassium drains away, the cell undergoes rapid repolarization, sending the voltage plummeting back down toward the negative baseline. These potassium channels are slow to close. The voltage actually overshoots the resting potential, driving the cell into a brief period of hyperpolarization before leveling out. This hyperpolarized state creates 2 distinct safety intervals. During the absolute refractory period, those sodium channels are physically plugged, making a second firing impossible, regardless of the stimulus. Shortly after, the plug resets. The cell enters the relative refractory period, where it can fire again, but only if the incoming stimulus is significantly larger than normal to overcome the hyperpolarized baseline. The pufferfish carries tetrodotoxin, or TTX, a specialized molecule that targets the voltage-gated sodium channel. If TTX enters the bloodstream, it physically obstructs the sodium channels. Without that initial influx of sodium, the entire electrochemical cascade is extinguished at the source, resulting in rapid paralysis. This voltage spike is highly localized. The neuron still has to physically move the signal down the entire length of the axon to reach the next cell. As sodium rushes into one segment of the axon, it diffuses laterally. That internal positive charge depolarizes the adjacent segment of the membrane, bringing it to threshold and opening the next set of voltage gates. The signal propagates forward in a wave. Because the segment immediately behind it is stuck in its refractory period, the signal cannot echo backward. To maximize the speed of this wave, mammals wrap their axons in myelin, a highly insulative fatty lipid sheath. This insulation is so tightly wrapped that ion exchange is completely blocked, except at small exposed gaps in the myelin called the nodes of Ranvier. This diagram illustrates saltatory conduction. Instead of a slow, continuous burn down the membrane, the electrical impulse rapidly leaps from node to node, massively increasing conduction velocity. In demyelinating conditions like multiple sclerosis, the immune system mistakenly strips away this insulation. Without it, the electrical signal dissipates, severely slowing down information transfer and causing widespread motor and sensory deficits. Myelin allows mammals to coordinate instantaneous motor responses without needing nerve fibers the width of tree trunks to lower electrical resistance. Once that signal reaches the very end of the line, the synaptic bouton, it hits a hard physiological boundary. The electrical impulse cannot bridge the physical gap to the next cell. This animated schematic shows the synaptic bouton. As the electrical wave collides with the terminal membrane, the mechanism shifts from electrical back to chemical. This final voltage spike dictates one last structural change. Voltage-gated calcium channels open wide, allowing calcium ions to flood into the intracellular space. Intracellular calcium is the mandatory trigger for the next step. The ions bind directly to vesicles, small membrane-bound packets storing chemical neurotransmitters, and physically push them toward the outer boundary. The vesicles fuse directly with the cell membrane, spilling their chemical payload out into the synaptic cleft. These neurotransmitters drift across the microscopic gap and slot perfectly into waiting ligand-gated receptors on the target dendrite, immediately initiating a new response in the postsynaptic cell. But a continuous signal is often dangerous. The synapse must be immediately cleared of neurotransmitters, either by enzymatic breakdown, reuptake carriers pulling them back inside, or passive diffusion. Weaponized nerve gas targets this exact clearing mechanism. By functioning as a potent inhibitor of acetylcholinesterase, the gas prevents the enzymes from breaking down the neurotransmitters in the cleft. The synapse functions as a precise biological gate. If the signal does not terminate, the resulting muscle paralysis leads to respiratory arrest. Zooming out from the microscopic synapse, these cellular mechanics aggregate to form the macroscopic architecture of the peripheral nervous system. At the systems level, sensory data from the environment travels up afferent pathways, entering the spinal cord through the dorsal root. Meanwhile, motor commands are sent out of the central nervous system down efferent pathways, exiting through the ventral root to innervate the muscles. If you step on a nail, sending that pain signal all the way up to the brain for cognitive processing introduces supraspinal latency. In a survival situation, those lost milliseconds result in severe tissue damage. This diagram outlines the biological solution, the monosynaptic reflex arc. An incoming sensory signal enters the spinal cord, synapses exactly once, and instantly triggers an outgoing motor command. During a standard knee-jerk test, the strike to the patellar tendon stretches the muscle. That stretch travels up the afferent sensory neuron, which fires directly onto the efferent motor neuron in the spine. The knee-jerk reflex allows the spinal cord to detect sudden stretch as a potential tearing injury, instantly contracting the quadriceps to protect the joint before the signal even reaches the brain. The nervous system translates microscopic ion gradients into macroscopic movement,